Welcome, AP Biology, to our Chapter 8 lecture on photosynthesis. You should be paying close attention during this lecture to everything that resembles cellular respiration, because if you understood cellular respiration, you're going to understand most of photosynthesis already. So one of my favorite candies is Skittles, and they're not wrong when they say, taste the rainbow, because through the process of photosynthesis, plants are able to harness light to make food. And as we learned in ecology, that producers are the bottom of our food web that support all the other trophic levels, we would have nothing to eat at all without light or refracted light rainbows helping to make food and energy. So we know that light is a source of energy, and photons of specific wavelengths of light are going to be used by specialized pigments in plants. So those pigments are going to absorb that light energy. And using that light energy, they're going to turn water and carbon dioxide into glucose or sugar. So think about your typical plant leaf is green. Which wavelength of light is going to be the best for performing photosynthesis? So if we think about what makes visible light, so there's all sorts of different wavelengths of on the spectrum of things we can't even see. So we can't see infrared or UV, things like that. But there's a very narrow range that our eyes can actually process, and that's the visible light spectrum. Now there's shorter wavelengths with high energy like violet and blues, and then there's longer wavelengths towards the oranges and reds that are much lower energy. And so this spectrum makes up all the colors we can see. So a green plant, what is it going to absorb the best? Well, remember, why do we see color? A surface reflects light that it can't absorb. And whatever gets reflected from that surface to our eyes is the color we see. So that means a green surface, like a green leaf of a plant, is going to absorb red light. It'll absorb orange and yellow. It'll absorb blues and purples, but it's going to reflect green. So if it's reflecting green, it's not going to be able to harness green light. So think about the colors of leaves. Typically they're green, but then they start to change in the fall, and autumn's beautiful because we have all this whole palette of reds and yellows and oranges. It looks like the forest is aflame. So what's causing this change? Is it changing what it's absorbing? Is it now reflecting something else? Well, if we look at the absorption spectrum, which is the spectrum of light and what gets absorbed, here we can see more. If it's a higher line, it's more absorbed by the leaf. There's different types of pigments. So chlorophyll, which is the classic green pigment that absorbs light, is going to, as you can see, not absorb green light very well. But it's going to do much better in oranges and reds. It's going to do much better in blues and purples, which is why we don't see green leaves as purple or blue or orange or red. But there's other types of pigments. There's different types of carotenoids and different types of chlorophyll. So let's look at some of these. What happens in the fall is there's less sunlight because the days are getting shorter, and leaves stop producing chlorophyll in response to this because they know there's not going to be as much sunlight. So chlorophyll is no longer being produced. It's no longer going to be present in the leaf. So those other pigments that were there all along are starting to be visible. So your anthromycins or reds, or your carotenes that make the oranges and yellows, are suddenly going to be visible now. And the light they are reflecting, which it's always reflecting, those leaves are always reflecting some red. They're always reflecting some orange and yellow. It's drowned out by the massive amount of chlorophyll. But in the fall, when the chlorophyll is gone, now it's the carotene and anthromycin's time to shine. And so we start to see those pigments that were there the whole time. So it's less that the leaves are changing color, and it's more that their dominant color just goes away. So let's talk about photosynthesis. And if we break down that formula, we're going to notice that 6CO2 plus 6H2O mixed with some light makes C6H12O6 and 6O2. Now you're going to notice this is the exact opposite of C6H12O6 plus 6O2 yields 6CO2 plus 6H2O plus some energy. So cellular respiration and photosynthesis are going to form a nice little circle. The reactants of one are the products of the other, and vice versa. And if we think about this in a reduction oxidation formula, that CO2 is getting reduced and water is getting oxidized. And all of our CO2 carbons are going to make that sugar. And you'll notice that 12 H's, 12 H's, 12 O's, 6 O's, 12 O's, oh, another 6 O's, so 18 and 18, it all adds up. So photosynthesis is going to be actually a two-part process. So you'll remember cellular respiration had multiple stages. Here we have two: the light reactions and the Calvin cycle, which can also be called the dark reactions. So let's explore in the chloroplast a little bit where this is going to be taking place. So just like mitochondria, chloroplasts were originally bacteria that were swallowed by the ancestors of plants, and instead of being eaten, they said, wow, these guys make energy really efficiently, or they make sugar really efficiently. Instead of destroying them, I'm going to protect them. And there's various structures inside to keep those stages separate. You can see there's these stacks of discs. The stacks together are called granum, but an individual disc is called a thylakoid. And that's where a lot of our light reactions are going to take place. The chloroplast has its own version of cytoplasm called the stroma, which has a really high pH, and that is going to be the site of the dark reactions, or the Calvin cycle. And these thylakoids are going to have a pigment that we were talking about, chlorophyll, along with a couple others, that are going to absorb solar energy. So a chloroplast has an outer membrane and an inner membrane, sort of like the mitochondria. There's thylakoids inside, which are sort of like their own organelles. They're these membrane-bound sacs filled with light-absorbing pigments. This is going to be where the light reactions take place. These are organized into stacks called granum. And then, as I said, there's the stroma, the aqueous solution filling the chloroplast. And then there's the lamella, these thin connecting bridges that kind of hold those thylakoids in place and maximize light exposure. So this perfect little once upon a time prokaryote is now the source of all energy, or most energy, in our ecosystem, I should say. So the first one is the light reactions, and as you can expect, they use light. As I mentioned, it's going to take place in the thylakoid membrane of chloroplasts. It's going to use water and light. Plus, we're going to see some of our old friends, electron shuttles. So we have NADP+, it's slightly different than just NAD+, and ADP. What's it going to make? It's going to make a waste product of oxygen, and we're very thankful for that waste product because it's the reason we can breathe and our atmosphere exists as it does today. NADPH, so a good electron shuttle, and some energy. We're going to get energy out of it. ATP, way to be. Nice. So, you're probably having flashbacks already to cellular respiration. That looks like an electron transport chain, and it is. There's three phases. There's photosystem 2 with P680 chlorophyll that absorbs light energy, getting electrons from water. It splits water into hydrogen ions and oxygen, and the leftover electrons get excited by light energy. In their heightened energy state, they go through an electron transport chain through the thylakoid membrane, producing some ATP along the way. We'll talk about how that works. Then it goes to photosystem 1, which is another protein involved in harnessing light energy, focusing it on P700 chlorophyll, which excites those electrons, and then that is used by an enzyme to make our electron acceptor NADPH. So let's break this down a little bit. But before I do want to say, why does photosystem 2 come before photosystem 1? Wouldn't it be photosystem 1, then photosystem 2? Well, the simple answer is scientists discovered photosystem one first, and so they called it photosystem. Then they discovered a different photosystem, so that since that was the second one they discovered, they called it photosystem 2. And then they realized 2 came before 1, and since they were already named, they said, just leave it as it is. So 2 comes before 1. So as a reminder, photons are waves that carry energy. And electrons can enter a higher energy state, which means they're able to be further away from that nucleus. They can enter an outer valence shell when they gain energy from those photons. They're called excited electrons. And that's going to power the processes in these light reactions. So chlorophyll molecules in photosystems are able to harness those photons and charge up or get those electrons in an excited state, which is going to give off heat. It can also give off its own source of light. So if you have pure chlorophyll and hit it with light and then turn off the lights, it can glow. So it's pretty cool. And there's these light harvesting complexes in the photosystem which bring in those photons, charge up those electrons, and get it ready for the electron transport chain. Water is a crucial component of this because you need a source of electrons. And so at that photosystem 2, water molecules, when electrons get excited and enter the electron transport chain, how you get more electrons is by splitting water into oxygen, which gets breathed out into the atmosphere, and hydrogen ions, which are going to be used in the electron transport chain. So remember, just like last time, electron transport chain, that flow of electrons is going to generate power. It's going to generate energy. And what are we going to do with that energy? Well, we're going to move hydrogen ions across a membrane against their concentration gradient into thylakoid space. So just like in the mitochondria, where we're moving it into that inner membrane space, now we're moving it into the thylakoid space. We're pumping hydrogen ions against their concentration gradient. Then, by simple diffusion through an ATP synthase, that's going to power some chemiosmosis for us to make ATP. So this is the exact same method as we saw in cellular respiration. The proteins and enzymes in the electron transport chain are a little bit different because they're different organelles from different evolutionary backgrounds, but we see the same ATP synthase, the same movement of hydrogen ions. So using the energy from the electrons to move hydrogen ions across and then let them fall back through ATP synthase. You can think of it as like pushing water up a hill to let it run down through a hydroelectric dam, run through that turbine to power making of energy. ATP synthase uses that just regular falling of hydrogen ions, and that's going to power ATP's production from ADP. Think of it like a dynamo spinning. So here we can see the whole picture here. Photosystem 2 gets excited by light. Water gets split into oxygen and hydrogen ions. Those hydrogen ions are utilized in the thylakoid space. Those electrons from water, when they get energized by light, go through the cytochrome complex of the electron transport chain to move hydrogen ions into the thylakoid space. So we have hydrogen from water and hydrogen getting moved across, filling up our thylakoid space. We're really high concentration here. Those hydrogens flow through ATP synthase to make ATP. The electrons at the end of the electron transport chain are going to get excited by light again in photosystem 1, and then we're going to reuse those high energy electrons to be accepted by NADP, our electron shuttle, and that's going to carry those over to the Calvin cycle. So we have ATP as a product of our light reactions going to the Calvin cycle, and NADPH going to the Calvin cycle. So now let's talk about our Calvin cycle. And in order to talk about Calvin cycle, we need to talk about a friend called rubisco. Rubisco is an extremely important enzyme that takes carbon dioxide carbons and builds a carbon molecule converts it into 3 carbon molecules called pGal that they can stick together to make glucose. And now this is going to require a lot of electrons, which is where that NADPH comes in. It's going to require some energy, so that's where ATP is going to come in. And so Rubisco is going to be a good friend here. One important thing we'll talk about a little bit later is Rubisco also is involved in photorespiration and can bind to oxygen just like CO2. And this can cause problems in some plants. And so we'll talk about an evolutionary way they get around that without wasting too much through photorespiration. So let's look at our Calvin cycle, also called carbon fixation. And we're in the stroma or the cytoplasm of the chloroplast. We're going to use 3 molecules of carbon dioxide for each turn of the Calvin cycle, 6 NADPH for each turn, and 9 ATP for each turn. And you'll notice that'll make 1 pGal, a 3-carbon building block for a sugar. Since sugar has 6, that's right, just like the Krebs cycle, we're going to do 2 full cycles. It'll release some NADP plus to send back to the thylakoids and some ADP and phosphate to send back to the thylakoids. So I'm going to kind of break this down real quick how this cycle works. CO2 comes in and Rubisco is going to bind that to a 5-carbon sugar. So you can see there's a 5-carbon sugar in there. Rubisco is going to fix that carbon into that to make a 6-carbon sugar. Unfortunately, this sugar is extremely unstable, and so it's going to break into 2 3-carbon molecules called pGal. Now, then, each of these molecules is going to take a phosphate from ATP to get converted slightly. Electrons are going to be donated from NADPH, and it's going to get converted again. And then one of those molecules that was made is going to be leaving to become sugar. The rest is going to stick around in order to continue this cycle. Remember, if everything leaves, you need more coming in than just carbon dioxide. So one's going to cycle back. So even though we made enough for a full sugar, one stays in. And if you notice the 1 CO2 molecule, maybe the CO2 molecule will stay in, maybe it'll get kicked out into sugar. So all of our carbon dioxide we breathe isn't necessarily going straight into sugar. Finally, regeneration, because a cycle has to be a cycle needs to be a cycle. We have to replace those 3 RUBPs that we started with. So we're going to take some of these G3P molecules that we have, and we're going to add some ATP and build 5 carbon sugars. So we're going to take 5 3-carbons and make 3 5-carbons, 15 carbon either way. And now we're ready again for 3 more CO2s. So here you can kind of see it added all up. We have those 3 Rubisco biphosphates that are coming in to 3 CO2 molecules. Those are going to make those 3 6-carbon sugars. They break apart into 6 3-carbon sugars. 3 of those are going to get, one of those gets kicked out. The other 5 stay in. And then we're going to regenerate our 3 molecules to then bind to carbon again. And that's going to be our cycle that we go through. Since we have to go through 2 turns to make 1 molecule of glucose, the Calvin cycle is going to make 2 turns. So that means we need a total of 6 carbon dioxides, a total of 18 ATP, and a total of 12 NADPH. Now this is a pretty good, even though we're investing 18 ATP, remember through cellular respiration, that sugar can get broken down into almost 38 ATP. So we're getting a lot more than we're putting in by making sugar. So it's a good investment in the long run. And remember, why don't we just use the ATPs that from the light cycle to power everything in the cell? Well, sugar has can make a lot more ATP for its size than a single ATP molecule. So sending all the ATP through the plant, there just wouldn't be enough space in the plant for all the ATP it needs to live. And so if you remember glycolysis, glucose gets broken down into 2,3 carbon sugars that ends up as pyruvate. One of those intermediaries is a P-gal, basically, a 3-carbon sugar. We're just going to reverse, once the products of the Calvin cycle are just going to reverse glycolysis to build those 6 carbon sugars. So G3P or P-gal is our sugar precursor, and with 2 of them, we can make that sugar. So Calvin cycle, we're putting carbon dioxide in, putting ATP in to make sugar, as opposed to taking sugar, kicking out ATP, kicking out ATP, kicking out carbon dioxide. So it's basically a reverse Krebs cycle. So I mentioned earlier there's different strategies because Rubisco can bind to oxygen as well, and that can speed up photorespiration, and plants can lose a lot of water that way because their stomata are open. So there's different strategies. So there's 3 types of plants we'll be talking about: C3 plants, just normal photosynthesis; C4 plants; and CAM plants. So C4 plants, things like corn and sugarcane that live in really hot temperatures. Because Rubisco is attracted to oxygen, it can impede carbon fixation, making it harder to make sugar with Rubisco constantly doing photorespiration. As a result of this, there's these special bundle sheath cells that take all the CO2 and store it there. And before it enters the Calvin cycle, it's going to store this CO2 as a 4-carbon molecule. So that's where you get C4 from that 4-carbon molecule. CAM plants, which are in super arid environments, really dry and hot, are going to use that C4 pathway to prevent excessive photorespiration. And then they also are only going to open their stomata at night. So they're going to store that CO2 in vacuoles, move it into the C4 pathway, and then enter the Calvin cycle. So CAM plants and C4 plants are definitely the alternates to that regular C3 photosynthesis pathway. So some important points I hope you learned: the visible light spectrum, how we see what we see, and what plants absorb versus what they reflect, what colors are going to be the best for plant growth, how the light reaction works. So remember, just like oxidative phosphorylation, only with light energy coming in, we still have an electron transport chain, we still have ATP synthase. The Calvin cycle, sort of like a reverse Krebs, CO2 is fixed into 3 carbon molecules. Those 3 carbon P-gals are then linked together to form glucose. And then finally, CAM and C4 plants have those alternate pathways for the Calvin cycle where they store CO2 as a 4-carbon molecule away from the oxygen so that there's not too much Rubisco photorespiration. And then they use it when they need it to make sure they can survive in their hot or arid environments. Hopefully you learned something about photosynthesis. Make sure to take the Kahoot quiz to study, and have a great rest of your day.